Hello everyone, I am Muhammad Ammar and today I am going to talk about the oogenesis. Oogenesis is the process in which ovum or egg is produced along with three polar bodies in a programmed series of division is known as oogenesis. Oogenesis occur in the outermost layer of the ovaries. As with sperm production, oogenesis starts with the germ cell called oogonium, but this cell undergoes mitosis to increase in number, eventually resulting in up to 1 or 2 million cells in the embryo. Now we'll discuss the mechanism of oogenesis. Like spermatogenesis, oogenesis is formed of three phases. The first one is multiplicative phase. In this, certain primary germ cells of germinal epithelium of ovary undergo rapid mitotic divisions to form group of diploid egg mother cells, oogonia. Each group is initially a cord and is called a tube of flugel which later form a rounded mass egg nest. And the second phase is the growth phase. The growth phase of oogenesis is of very long duration than that of the spermatogenesis. During growth phase, one oogonium of egg nest is transformed into diploid primary oocyte while the other oogonia of the egg nest form a single layer nutritive follicular epithelium around it. The structure so formed is called the primary follicle. Later, each primary follicle gets surrounded by more layers of granulosa cells and changes into secondary follicles. Soon, secondary follicles develop a fluid-filled antral cavity called the antrum and is called tertiary follicle. It further changes to form graphene follicles, so not all the oogonia develop further. The growth phase also involves the increase in size of oocytes, uh, 2000 times in frog, 43 times in mouse, 90,000 times in Drosophila, and 200 times in hen and 200 times in human females. By the formation and accumulation of yolk, the formation and accumulation of yolk is called vitellogenesis by a special mitochondrial cloud lying close to the nucleus called yolk nucleus. The nucleus becomes bloated with nucleoplasm and is called germinal vesicle. A thin white line membrane is secreted around the oocyte. Increase in the number of mitochondria, amount of endoplasmic reticulum and Golgi bodies. The formation of Lamprecht chromosomes in fishes, amphibians, reptiles, birds, insects, etc. for rapid yolk and synthesis. Gene amplification or redundancy of ribosomal RNA genes for rapid synthesis of ribosomal RNA. And the third phase is the maturation phase. It is characterized by meiosis. In this, the diploid and fully grown primary oocyte undergo meiosis 1 to form two unequal haploid cells. The smaller cell is called first polar body, uh, the podocyte, and has very small amount of cytoplasm. The larger cell is called the secondary oocyte and has a bulk of nutrient-rich cytoplasm. Both of these are haploid and each has 23 chromosomes. The secondary oocyte undergo meiosis II to form two unequal haploid cells. The smaller cell is called the second polar body and has very little of cytoplasm, while the larger cell is called otid. It has almost whole of the cytoplasm and differentiates into an ovum. Meanwhile, the first polar body may divide into two. Soon in oogenesis, a diploid oogonium forms one haploid ovum and two or three polar bodies, while in spermatogenesis, a diploid spermatogonium forms four haploid sperms. The primary function of formation of polar bodies is to bring haploidy but to retain the whole of the cytoplasm in one ovum to provide food during the development of zygote to form an embryo. The number of ova is reduced with the ability of the female to bear and rear them. In the most of the organisms, including human female, the ovulation occurs at secondary oocyte stage, in which meiosis I has been completed and first polar body has been released. And the meiosis II is completed only at the time of the sperm entry. These were three phases of the oogenesis: the multiplicative phase, growth phase, and maturation phase. Now we'll discuss its significance. It produces haploid ovum by releasing two or three haploid polar bodies. Most of the cytoplasm is retained in functional ovum, and variation may appear due to crossing over during meiosis I. It proves evolutionary relationship. So this is the schematic diagram of oogenesis, in which the oogonium develops into oocyte by mitosis, and then this oocytes converts to a polar body and two oocytes which are haploid, and then the meiosis occur in the oocyte and it forms further one polar body and a otite, and then the otite develops to a ovum. Thank you so much guys. This was all about oogenesis. If you like the video, please subscribe my channel for further such educational and recreational videos.